So now that we've really covered the main basis for organic molecules and their structure within the cell, we're going to be moving in and focusing specifically on cell structure and function, and how those macromolecules start to become processes within the cell. So the driving questions for this section is going to be what structural features are shared by all cells and what is the differences between prokaryotic and eukaryotic. So we've covered some of this before, but it's good to sort of go through it as a refresher, especially in terms of now that we're talking more and more about structure. Um, Second, how do solutes and water cross membranes? What determines the direction of movement of solutes and water and in different situations? Next, we want to talk about a little bit about how antibiotics target bacteria and what situation is antibiotic therapy indicated. And lastly, what are some key eukaryotic organelles and their functions? So we're not going to be going through all of these, but we are going to be looking at some specific ones that really have to do with packaging and understanding the macromolecules that are involved in cellular biology. So your book chapter in this section sort of explores these questions through the discovery of penicillin. So penicillin is a fungus, and that Alexander Fleming observed was capable of killing many different types of bacteria. So he actually discovered this by accident, where he left some cells cultures out overnight, and came back and discovered there was this white fungus growing on the plates, and he noted that several of the bacteria that were on these plates had essentially disappeared because of the presence of this fungus. This is actually a comic strip that was created in the 1930s that was honoring different scientists of the age. So I thought I'd include that here. So this is just more specifically looking at what happened on his petri plate. So the fungus that actually was able to disrupt the bacteria is actually the fungus that is very commonly found on oranges, where you see that white fungus that eventually turns to green. Um, and basically, this was sort of just a fortuitous event that happened. It was essentially random, where we saw that bacteria colonies at locations far away from the mold were stable, but were not able to form sort of near this mold. And from that penicillin fungus, we were able to derive one of the most important antibiotics that is still in use today. Um, and so we've discussed previously, when we were talking about antibiotic resistance, that essentially antibiotics are just a chemical that can slow and stop the growth of bacteria. And often these antibiotics are naturally produced by other living organisms. So, as I said previously, the penicillin is derived from Penicillium notatum, that fungus. Um, so when we start to think about antibiotics and the presence of cells, we have to start thinking in terms of cell theory. And that essentially is that all living things are made of cells, and that every new cell comes from the division of a pre existing cell. I'm going to have you guys watch another video looking at specifically the history of this cell theory, because while it sounds fairly intuitive to us, it really wasn't. Really was a novel idea of its time, and an understanding that cells were that all living things, not including plants, animals, protists, everything all life that we are aware of is made up of individual cells. And then secondly, that cells come from pre-existing cells. There are a lot of thinkers at the time that sort of thought that cells just sort of came into existence and didn't come from some other pre-existing cell. So this was a really revolutionary theory for its time. And ultimately, we ascribe to the theory that all living organisms are made up of cells, from single-celled eukaryotes and algae to amoebas, bacteria, to human muscular tissue, and aquatic plant tissue, and molds and fungi. Again, all of these different forms of life and clades of life, which we discussed in our previous section, all depend on this basic subunit of the cell. And previously, we discussed that there's two main types of cells. There's prokaryotic cells and eukaryotic cells. And the main thing that distinguishes them is this lack of an internal membrane bound organelles. And also, prokaryotic cells are much smaller than eukaryotic cells. And you can see here that prokaryotic cells lack this interior nucleus or these endoplasmic reticulum, mitochondria, but you can see that they have a cell wall as well. And so these are the features that that are specific to both prokaryotic and eukaryotic cells, and then there are a few different organelles that are different from them. And you can see that these pictures are particularly illuminating, where you can see just the free-floating nucleic DNA versus where we have this distinct nucleus that contains the DNA in the eukaryotic cell. So one of the key features of prokaryotic cells is that presence of that cell wall. And when we're thinking about bacteria, there are two major classes of bacteria. There are gram-positive bacteria and gram-negative bacteria. And gram-positive bacteria, that cell wall with that layer of peptidoglycan, can retain this gram stain, versus gram-negative bacteria, the cell wall of the peptidoglycan is surrounded by a lipid membrane that does not retain that gram stain. And it prevents, and in the case of gram-negative bacteria, it prevents penicillin from reaching to the peptidoglycan underneath. So here we have a structural difference that is enabling one class of antibiotic to be able to act on a specific cell compared to another. So this is just like big main groups of bacteria, and ultimately these groups depend on the different antibiotics will work at different efficacies based on whether or not there are gram-positive or gram-negative bacteria. So like I said, penicillin is effective against these gram-positive bacteria, but not against gram-negative. So while it was this huge boon to society for penicillin, we see these war posters, thanks to penicillin, he will come home. There are still other methods of killing bacteria are needed. For example, streptomycin can interfere with prokaryotic ribosomes and leaves the eukaryotic ribosomes unaffected. So there are lots of different types of antibiotic that start to react with the different structures of prokaryotic cells, and leads to in the case where we are now, we have lots of bacteria that are antibiotic resistant, which has led to a lot of the processes that we discussed in our previous lecture. And a lot of really what is making a bacteria resistant or bacteria susceptible to different types of antibiotic depends a lot on the structure of the cell wall. And that's what we're going to start to really get into in our next lecture. So I'll see you in a little bit. All right. Thanks. Bye.